All right, welcome everybody to Chapter 14 review. This is your second to last video as we make our way to the end of the course. We will have Chapter 15 review following this one. For Chapter 14, as it always, this is a non-comprehensive review, but I will help you focus in on some areas that you could concentrate on when it comes to Chapter 14. Electroencephalograms and associated waves, meaning like alpha waves versus beta versus gamma. What are the waves? What's an electroencephalogram? How can they, what is it about the waves when it's detected on the machine or on that device? Lobes of the brain and their function. Everything from the frontal lobe, occipital, parietal, temporal, even underneath the limbic system and all the other systems underneath as well, or within the brain themselves. What are their functions and where are they located? Association areas. Again, association areas, as you know, are like Broca's area or Wernicke's area. You should be familiar with those areas that are for hunger or for speech or sex drive, blood pressure. So when it comes to the lobes, when you study the lobes of the brain and when you study the association areas, you kind of can study them together because you do need to know the functions of each lobe in relationship to how does each lobe communicate with each other, and usually it's through the association area. Embryonic development. Okay, so looking at mesencephalon, myencephalon, metencephalon, diencephalon. What does each part represent as an embryo, and what will it develop into as the human or creature evolves? We have the cerebellum and their function and its function. Pretty straightforward. Okay. Basal nuclei, location and function. When it comes to the basal nuclei, where are they located? When you do a cross section or a frontal section of the brain, can you see the basal nuclei? And if you do, well, then what is the function of those structures? Cerebrospinal fluid, CSF. What is it? Where is it located? Obviously, it's located around the brain and down the spine. Okay, but also more particularly, where is it produced and what's its function? Understanding the production of CSF will also help you understand where it's located and also its function, but its production is also based on some type of glial cells that you'll have to understand and know as well. So where is CSF located? Think about ventricles in the brain, lateral versus the third ventricle, so forth and so on. And with that, you should be able to put together where the CSF is located and its production. All the meninges, all the great meninges from the dura to the pia to the arachnoid. Obviously, it's not in order, right? So dura mater, arachnoid, and pia. Make sure you know where they're located, how they work, and their function. Also understand that the the meninges also extends into the brain. Okay, you have the tectorials, the tectums, and all the other parts of the meninges as it branches up into the skull and also helps divide the brain a little bit, right? It helps give some separation between the cerebellum and the cerebrum. Or there is also some in between the hemispheres. So when you did your dissection in lab, I'm not sure if they did this year, but some of them had the dura mater on top, and then some of the tectums will actually be seen between the different lobes of the brain as well. So be familiar with all the meninges, location, types, and functions. Of course, cranial nerves, number, location, and function. Pretty straightforward. You have your mnemonic, hopefully, of them in order. You should know which are sensory, you should know which are motor, which are both. Know them in order, 1 through 12, and of course their function. Know the sensory functions and the motor functions when it comes to the cranial nerves, and both for that matter. Like, those that have a lot of functions, like the vagus nerve as well. You should be familiar with all those functions. Some terms that you may want to look into: corpus callosum. Where is it? What's it do? How does it do its job? Arbor vitae. Well, it's kind of like the corpus callosum. I spelled corpus callosum wrong. I apologize. It's not corpus callosum. It's corpus callosum. So basically, where are they located? and what's their function? Purkinje cells, what are they? Lateral sulcus, where is it? Medulla oblongata, limbic system, pre and post central gyrus, reticular formation. So you see where I'm going? I'm kind of going over some of those hills and valleys, right? Those sulci and gyri of the brain itself. So be familiar with the folds and within it of the brain there. Neural tube formation, that kind of comes into play with the neural folds. Understand that from embryonic development. Going back to our list above there with the reticular formation, you should be familiar with stuff also as Wernicke's area and pyramidal cells. Okay, or Purkinje cells and what they're related to. So in that paragraph, they have Purkinje cells, but you also should also have pyramidal cells in there as well. Association tracts, we did that one already. Okay. Good. Ventricle location. So in the brain, lateral ventricle, third ventricle, other ventricles, be familiar with them. What does it do? Where they locate, know the structures in front of or perhaps behind the ventricle, so you get a better idea of where they're located. Different types of memory. Right? Long-term versus short-term, declarative memory versus conditional reflexes or procedural memory, which is really a real memory that we're working with. But be familiar with something like declarative memory. The textbook does cover some different types of memory. Be familiar with those types, and you should be fine with that. We did diencephalon, precentral gyrus, postcentral gyrus, I already said that. CSF functions, said that. So I will repeat myself as we go through this. Primary somatostatic cortex. Right? We have that nice homunculus concept. Where are different parts of the body sensed, if you would, when it comes to the homunculus and different sensory inputs? We said ventricles already, right? Cerebral aqueducts, third ventricle, lateral ventricles, gray matter versus white matter. I think that's coming up before we get there. Mesoderm versus ectoderm versus endoderm. This is kind of going back to a little bit of the embryonic development, if you would. So make sure you know who is developed from who, right? Ectoderm, mesoderm, endoderm. So they're not in order. There's really no order, but if we go from outside, middle to inside, it should go ecto, meso, and endoderm. So be familiar with those concepts. Good. There is going to be a question, if you're lucky enough to get it, there will be a question of tracing CSF flow through the ventricles, right? So it's going to be starting at point A and moving you through point B through C, D, E, F, so forth and so on. Be familiar with how does CSF flow? Comes out of the blank and goes around the brain through this ventricle to this ventricle, right? Ependymal cells are involved. Earlier in the review, I was talking about these tectums. It's also called like the falx cerebri, okay? The falx or tentorium cerebri. I call them, what I said is the same thing, but I was just shortening it up. So going back to the concept of dura mater, arachnoid, and pia mater, and how this dura-type material extends up into the brain and helps separate out the cerebrum from the cerebellum, we have what's called tentorium cerebri, tentorium cerebelli, falx cerebri, falx cerebelli, and falx is F-A-L-X. Be familiar with those as well. Right? So kind of get the large concepts of those as well. Trace the flow of CSF, I already said that. So if we start you off the lateral ventricle, you could go from there. Or maybe we'll start you off at the subarachnoid space. But it's multiple choice, so you should be able to trace it right through with no problem. Blood-brain barrier. What is a blood-brain barrier? Who are the cells that help make up the blood-brain barrier? What could pass the blood-brain barrier? What can't pass the blood-brain barrier? As for those of us who understand, who may go out and have a good time and with a designated driver, alcohol does pass the blood-brain barrier. Some chemotherapy medications do not for those with brain tumors. So what a lot of scientists do, or drug companies would do, they'll take the chemotherapeutic medicine or drug, attach it to a glucose molecule, and we know glucose does cross the blood-brain barrier because that's what the brain uses as fuel, and helps to bring in the chemotherapeutic drug to the brain by using a glucose molecule. So be familiar with the blood-brain barrier, the cells involved, and how it works. Right. So we know that astrocytes are one of the major players of the blood-brain barrier. So are ependymal cells, believe it or not. Right. So it's not just I'm going to go with astrocytes and microglial cells, not ependymal cells. So something to look up. I may have to change that test question. Hint, hint. Okay. Let's see. Gray matter versus white matter. Why is gray matter gray? Why is white matter white? Myelin versus unmyelinated fibers. For example, a portion of the gray matter in the pons, medulla, and midbrain is called a reticular formation. So what is the reticular formation? I don't have that on my review, so be familiar with the reticular formation and how it relates to gray matter. Some other nuclei that you should be familiar with, location of function, is the paraventricular nuclei, suprachiasmatic nuclei, and the preoptic nuclei. Be familiar with those three. Very specific. As you can see, my reviews are pretty specific. I pretty much have the exam open right here. So I'm trying to get you in the right direction here, okay? Know your numbers of cranial nerves. Know your association areas. Okay, so as you can see, it is it's a big chapter. The nervous system is a big chapter. All nervous systems are big chapters. So be familiar with everything we just talked about. So if I can find anything else that's important. It's all important, but I pretty much touched upon some of the concepts that you're going to run into. Parkinson's disease. Be familiar with that. Alzheimer's. Meningitis. A couple of things I left out. I apologize, but gets the point across. Different nuclei. So that's about it. Okay, so go over the review again. If you have to, definitely study chapter 14, as it is going to be part of one of the 2 chapters that you're responsible for on the exam. And other than that, any questions, concerns, or comments, please feel free to give me an email, and I'll get right back to you. So good luck with everything, and I'll see you in the next review.